Good morning, everyone. So we will start today in the morning our presentations. So it is my honor today to present you, to introduce you to Dr. Makeda Tekle-Smith, visiting us from Columbia University. Her chemistry path began earning bachelor degree in chemistry at Pomona University in 2014. After 5 years, Makeda obtained her Ph.D. with Professor James Leighton at Columbia University. There she developed new methods to construct asymmetric carbon-carbon bonds and applied these technologies to the total synthesis of non-aromatic polyketides natural products. Following that, Makeda conducted her postdoctoral research with Professor Abigail Doyle, first at Princeton University and then at the University of California, Los Angeles. There she the work focused on generating and harnessing reactive radical intermediates through photoredox catalysis. In 2022, she returned to Columbia University as assistant professor and started her own research group. Leading the group, Makeda continues to push the boundaries of knowledge, exploring fundamental principles of physical organic chemistry within the realm of asymmetric synthesis. That means elevating the reactivity of unconventional chiral motifs, advancing foundational understanding of chiral structures, effects, and creating enantiopure substances, which then can be applied in pharmaceutical development, material sciences, and advancement of sustainable synthetic methods. So please join me welcoming Makeda Tekle-Smith to present her work about alkynes and photoredox catalysis. So.
Thank you so much for that kind introduction and for the invitation to be a speaker in this fantastic conference. I've had such a wonderful time meeting all of the scientists and the students here. It's been really, truly a wonderful week, and it's such an honor to be able to speak with the other esteemed speakers in these sessions. So I'm going to be telling you today about a variety of different projects in photoredox catalysis. And I really love to start with this slide, which is a variety of single crystals that I grew of iridium photocatalysts. And here we were looking at how we can grow single crystals of these iridium photocatalysts bound to anions to see how these types of anions can affect the photoredox properties of these catalysts. And I think that that's really one of the things I've found so interesting about this field is that we draw these catalytic cycles and we think about these types of electron transfer processes as being very discrete. But they're quite complex and are dictated often by the types of intermediates that we have in solution at a time. And so these processes are incredibly complex and deserve an intense amount of study. And so to step a little bit further back, I just wanted to describe a little bit about why we are interested in photocatalysis and why we think that it's an important area of interest to study. And so one of the motivations that my group and other groups in the field have is to solve this, or to try to solve this demand for energy that's derived from these chemical syntheses that are increasing at an ever-increasing rate. And so the big challenges for society is thinking of how we can supplement fossil fuels with clean, renewable resources. And just to take a smaller look into this from a US perspective, chemical industries use around 29% of the total energy consumed in US manufacturing. And so if we can, as chemists, find novel ways of developing renewable resources in synthesis, we can really improve the energy consumption that the chemical industries use. And so how do we harness light energy for chemistry? One of the ways in which we can do this is with photoinduced electron transfer, where upon excitation of a catalyst, we can generate both a reductant and an oxidant and develop this into photoredox catalysis or solar energy conversion. And so I'm going to be really thinking about photoredox catalysis in this talk and how we can apply this in chemical synthesis. And I think one of the fantastic examples of using this to make challenging bond transformations is in metallophotoredox catalysis, where you're using the fundamental steps in photoredox catalysis of oxidation, reduction, and energy transfer, and combining this with transition metal catalysis, elementary steps of oxidative addition, transmetallation, reductive elimination, to be able to design a plethora of new mechanisms. For example, decarboxylation, CH functionalization, cross-coupling to generate CSP2 and CSP3 bonds. This was nicely highlighted by Dave McMillan's group, Abby Doyle's group, and Gary Molander's group in 2014. And so one of the big industries that these types of bond transformations are useful in is in the drug discovery process. And almost 50% of FDA-approved drugs in the United States are derived from natural products. So when we're thinking about designing these types of new reactions, we're taking inspiration from these natural products in thinking about the types of bonds that need to be formed and the routes in which we can most efficiently form these types of natural products. Some of the classical examples of natural products that we see are shown here, such as quinine, an anti-malarial, Taxol, an anti-cancer drug, lovastatin, which is a statin for cardiovascular health. And these natural products are derived from nature, but often the natural products, compounds that we find in nature can't be accessed simply from the material where we found these compounds. And so this is really where total synthesis comes in. We need synthesis to be able to produce large amounts of these compounds without depleting the natural resources of our world. And often the compounds that we find in nature, while they give us inspiration for targeting specific pathways for pharmaceutical development, we don't want simply the compound that we discovered. We want to be able to derivatize this to reduce side effects in human health, or to get at a better pharmacokinetic profile of these drug intermediates. And this is where my research is really focused on, is developing new reactions to be able to more efficiently make these drug-like molecules. And so the interest that my group has been focused on and my career path has gone through is thinking about how we can design new catalysts and reagents to enable these reactions to be developed. And the types of reactions that we're targeting are inspired by these natural products. And then we apply these to pharmaceutical development as well as materials development and developing new biomedical tools. And so two of the projects that I worked on as a postdoctoral fellow in Abby Doyle's lab are highlighted here. These are both using photoredox catalysis, one using a metallophotoredox platform coupling an iridium photocatalyst with a nickel catalyst and to generate these types of CSP2, CSP3 bonds, where you can take an aryl chloride and a common solvent, such as trimethyl orthoformate, as your precursor to a methyl radical for methylations of these aryl chloride starting materials. But the main focus that I'm going to be talking about today is CH functionalization, how we can activate these strong CH bonds to generate a variety of different products in an incredibly efficient manner, again, using photoredox catalysis. And so when I think about developing a new method using a radical process, I think about it in two steps. One, how you generate the radical in a mild, functionally group-tolerant way. And the second process, how do you intercept this radical through a radical trap or through conversion of that radical to another sort of intermediate, like a cation, for example. And to get at these types of high-value transformations, like methylations, fluorinations, esterifications. And so when thinking about picking a precursor that's ubiquitous in chemical structures, a CH bond is one of the first things that we think about. They're everywhere. The number of substrates that you can use is immense. And so if you can access these types of CH bonds to generate radicals, the synthetic capabilities that you have is quite incredible. And so we can think about how we can more efficiently couple two different partners to generate drug-like molecules, like shown here, from common intermediates, like these arenes. Additionally, if you have a complex structure already synthesized, like these types of drug intermediates shown here, you can generate libraries of these derivatized compounds in a single step. But this activating CH bonds is incredibly challenging because of the strength of these types of bonds, as well as in terms of thinking about selectivity of these types of transformations. So in just one of these compounds shown here, there are many different types of CH environments. And so we need to think about designing new catalysts and reagents that can predictively activate one of these types of CH bonds in the presence of all of these others to be able to develop a useful type of transformation. And so some tactics for breaking CH bonds that have been used in the literature previously are transition metal insertion, hydride abstraction to generate a carbocation intermediate that can then interact with nucleophiles to generate CX bonds, as well as hydrogen atom transfer, which is going to be the main focus of my talk today. Hydrogen atom transfer needs a hydrogen atom abstractor to activate that type of CH bond to generate this hydrogen atom transfer process. And some of the common electrophilic hydrogen atom abstractors I've shown here, such as quincuridine, bromine or chlorine radicals, and other nitrogen and oxygen-centered radicals, are what typically is seen in the CH functionalization literature. And when selecting a hydrogen atom abstractor to do a CH functionalization step, you want to think about a few different parameters. The bond strength that you're forming with your abstractor compared to the bond strength that you're breaking, as well as the polarity of the radical that your hydrogen atom abstractor is. So radicals can be either electrophilic or nucleophilic comparatively. And so you can think about this as you would for two-electron chemistry, where you have a nucleophile reacting with an electrophile. You can similarly have an electrophilic catalyst, such as quincuridine shown here, and this is polarity matched to abstract from more electron-rich CH bonds, even in the presence of weaker CH bonds, for example. So there's many different factors in which control these types of hydrogen atom transfer reactions. And because the majority of known hydrogen atom abstractors that are seen commonly in the literature, these nitrogen or oxygen-centered radicals, are more electrophilic in nature, they're going to be abstracting a certain type of CH bond preferentially. And so one of the motivations that we had when thinking about developing a CH functionalization method was thinking about developing new nucleophilic hydrogen atom abstractors, which there are several of in the literature that have been previously explored. But it's quite a narrow number compared to the variety of electrophilic hydrogen nanoabstractors that are commonly used. And these, you might imagine, are more challenging to think about designing. Because doing, for example, a carbon-to-carbon hydrogen atom transfer is going to be less favorable than a nitrogen-to-carbon hydrogen atom transfer for many examples, especially in not an intramolecular fashion. But the benefit of this is that you can access different types of CH bonds. So for example, the majority, if you have a compound like this, the majority of electrophilic hydrogen atom abstractors will abstract this blue highlighted CH bond, whereas nucleophilic hydrogen atom abstractors are preferential for this alpha carbonyl CH bond. So we thought along this radical felicity scale that methyl radical might be a preferential hydrogen atom abstractor that we could think about designing a reagent for to do this rare CSB3 to CSB3 HAT step and think about how we can explore a different type of chemical space for this CH abstractor. And so the reagent that we thought would be positive in terms of generating this methyl radical would be this redox active phthalimide ester shown here, where this phthalimide ester can accept an electron from a photocatalyst to generate this radical anion that can then decarboxylate to generate phthalimide anion, carbon dioxide, and methyl radical. And so why we thought that methyl radical would be an interesting mildly nucleophilic hydrogen atom abstractor is that it will generate methane upon hydrogen atom abstraction, which has a very strong bond dissociation energy. This phthalimide ester is a very bench-stable methyl radical precursor, and it's incredibly easy to make. It's a dump and stir reaction. And it's also lasts for a very long time, so it has a very strong shelf life. The reduction potential of this phthalimide ester is also quite low, so a number of different photocatalysts should be able to do this initial electron transfer step. And it has a non-nucleophilic byproduct of methane being generated. And so the catalytic cycle that we proposed in this work, which was done in collaboration with, at the time, a fantastic graduate student who's now a postdoctoral fellow at Harvard, Dr. Isabel Liebler, we proposed this type of catalytic cycle, where you could take an iridium photocatalyst, excite it to be an excellent reductant that could do this single electron transfer reduction step shown here, that can then decarboxylate to generate methyl radical. This methyl radical can perform this intermolecular HAT step with our CH partner of interest to generate a radical intermediate. After this reduction step, you are at an iridium-4 intermediate, which will now perform as an oxidant to do another single electron transfer, generate a carbocation while turning over your catalytic cycle. And then this carbocation is primed to do classical SN1 type chemistry with various nucleophiles to generate your bonds of interest. And so the type of bonds that we initially wanted to attain are CF bonds. And why we wanted to target this specific type of reaction class is because of the importance of CF bonds in pharmaceuticals and in imaging technologies. Specifically, installing F18 on compounds is incredibly important for generating new PET tracers for imaging applications. And we wanted specifically to develop a nucleophilic fluorination strategy that could be applied to F18 chemistry because using fluoride instead of electrophilic fluorination sources is quite preferable in radiolabeling applications. But using fluoride is quite a challenge in various fluorination chemistry because of the attenuated nucleophilicity of fluoride, as well as in many of these fluoride sources, the solubility of these reagents is quite poor, and fluoride can also act as a base often rather than a fluorination source. And so developing nucleophilic CH fluorination methods is very challenging, and there's very few examples of these types of CH fluorinations in literature. And so we wanted to add to this field. And so the model system that we started to explore this chemistry with was using diphenylmethane as our CH partner, shown here, and triethylamine HF as our fluorination source, and N-acetoxyethylamide as this methyl radical source, with an iridium photocatalyst in pivaloNitrile as our solvent. And we were able to see successful fluorination of this CH partner and an 88% yield under these conditions. Sorry. And so we see a dependence on this, of the success of this reaction on the photocatalyst that we're using specifically. And we found that this monofluorinated photocatalyst that had a high reduction potential as well as a high oxidation potential was preferential for this type of chemistry. As we decrease the reduction potential of our photocatalyst, we see a decrease in yield, as well as if we have a decrease in the oxidation potential, we also see a decrease in yield. And this is indicative of the mechanism that we're proposing, as both of these electron transfer steps, both to generate the radical as well as to generate our carbocation, are critical in this type of chemistry. Just to highlight a few of the reactions, the substrates that we can access in this type of chemistry are shown here. We can fluorinate both secondary and tertiary systems, as well as a variety of heterocycles. One of the limitations of this chemistry is electronic in nature, so more electron-rich functional groups will lead to higher yields in this reaction compared to electron-poor functional groups. And this is also indicative of the mechanism that we're going through. And we're able to fluorinate a variety of known drugs as well that are commercially available. But I would like to put a caveat here. This is not, I would say, a wide substrate scope. It is, I would say, pretty typical for CH functionalization chemistry, but we really wanted to evaluate based on what is known fluorination space, how this type of chemistry maps onto this. So there are a number of drug molecules that have been developed that have these CsP3F bonds and proposed structures as well. And the Doyle group has been developing nucleophilic fluorination chemistry for some time. And so we have a library of the types of compounds that we've been able to make with our various nucleophilic fluorination reactions. And so we wanted to evaluate how the types of CF bonds that we can form fit onto what is known chemical space. And so one of the things that we did is did a database search of all CF3F bonds in various structures. And then we did a hierarchical clustering of this reaction space to generate these 5 clusters of various types of CF bonds in the known database, which is what the database that we used was Reaccess. And what we saw from the types of CF bonds that the Doyle group could produce with our nucleophilic fluorination methods is that clusters 1 and 3 are highly overrepresented of the types of CF bonds that we can form. And so one of the questions that we're asking ourselves are, our new methods or redesigned target scopes needed to reach this sort of dark chemical space shown here. So these dark blue dots are the chemical compounds that our substrates, our scopes can access, and all of this gray area is what we don't have as representative in our reaction scopes. And just to give you a sort of sense of what these types of compounds are, these are what is overrepresented in our scopes, clusters 1 and cluster 3. And you can see that in clusters 2, 4, and 5, these are much more complex chemical architectures. And so I think the takeaway that I have from this sort of data analysis of the known CSP3F chemical space is that we're not doing a great job of selecting substrates or evaluating really the scopes of these reactions in an effective way. And so this is what I'm hoping that we will get to in the future is being able to use these sort of data science tools to build more effective scopes that can track this type of dark chemical space and really see how diverse and how robust our reactions are. And if our reactions can't access a specific area, to then design reactions for that specific dark chemical space. And specifically accessing these types of, these clusters 2, 4, and 5. And I would say that the Doyle Group's scopes and the types of reactions that we develop are pretty typical of what's seen in the literature. But getting back into the mechanism of this reaction, we wanted to explore if what we proposed initially that's giving this fluorination is actually accurate. And so one of the main questions that we had was, are we generating this methyl radical? And is it the active hydrogen atom abstractor in this reaction? It could just simply be initiating a radical cascade mechanism to generate another active species that's abstracting this hydrogen atom. So to start exploring this, we first wanted to ask, are we generating methyl radical at all? And the way in which we did this is via radical trapping experiments. So if we add in a stabilized radical, for example, tempo, into this reaction, we see an attenuation of the fluorinated product itself, and we see trapping of methyl radical within this type of tempo adduct shown here in a 32% yield. Additionally, if we add in an alkene instead of an alkane as our substrate in this type of reaction, we see almost quantitative trapping of this methyl radical and a 1,2 now carbofluorination in almost quantitative yield for this reaction. Additionally, so this shows that we're generating methyl radical, but it doesn't say whether the methyl radical is the actual active species that's abstracting this hydrogen atom. And so to explore this, we performed photo-NMR reactions, where we set up the exact same reaction in an NMR tube and inserted a LED into this reaction to irradiate the NMR. And then we were able to observe this reaction progress in real time. And so in this first top reaction shown here, we can see methane being produced as the reaction is going. And so we know that methyl radical is being generated and that it's abstracting a hydrogen atom to some sort of efficiency. However, I think the more important reaction that we performed was when we introduced deuterated diphenylmethane into this reaction. We were able to see deuterium incorporation into our methane and see deuterated methane increase over the reaction progress. And so I think that this is quite good evidence that methyl radical is doing the hydrogen atom abstraction on our substrate of interest. One of the benefits, I think, of this mechanism as well is that we're performing this radical polar crossover step to generate a useful carbocation intermediate in a mild way. Simply going from a CH bond to a carbocation typically needs quite harsh conditions under typical two-electron chemistry. And so doing this in this sequential single electron transfer process is a mild way to get a useful intermediate that we see in a number of classical organic chemistry reactions. And so we've just shown one sort of transformation that you can achieve, this CF bond formation. But in theory, you can swap out fluoride for any sort of nucleophile in this chemistry and see other types of bonds being formed. So we wanted to explore this a little bit further. And we were able to replace fluoride with a variety of different alcohols and generate carbon-oxygen bonds as well to varying degrees of success in this reaction. We can also see successful chlorination and azidation and thioetherification as well. We can also do Friedel-Crafts type alkylations using these electron-rich arenes as nucleophiles to generate carbon-carbon bonds in this type of transformation. So this project, I think, shows nicely how we can use photoredox catalysis for CH functionalization, converting a single CH bond to a functional group of interest, using a mildly nucleophilic hydrogen atom abstractor, which has shown to have some unique selectivity platforms than compared to traditional electrophilic hydrogen atom abstractors. But this is a specific single transformation where we're converting one CH bond to one functional group. And I think that this is pretty typical for a lot of CH functionalization chemistry, where the challenges that you're solving are getting selectivity for one specific type of CH bond and trying to limit side reactions and unproductive chemistry. One of the challenges that my independent group has really taken on and focused on is thinking about how we can take unactivated alkanes and functionalize them in difunctionalized products. So functionalizing two types of CH bonds in a single step from a very common substrate classes. And this type of difunctionalization chemistry introduces a variety of new challenges in this field. And therefore, we need to design new catalysts and new reagents to access these types of difunctionalized products. One of the key differences between a single functionalization method compared to a difunctionalized method is thinking about the type of selectivity that you're going to get between these types of CH bonds. So whether you want a 1,2 difunctionalized product or a 1,3 difunctionalized product, et cetera. And so this is one of the challenges that my group has really taken on, is thinking about how we can design new mechanisms and new ways of thinking about this type of difunctionalized chemistry. And so one of the ways in which you can think about this is generating these types of di-radical intermediates that can do dual CH abstraction and generating alkenes that can then map onto classical difunctionalization of alkene chemistry. Additionally, you can think about sequential hydrogen atom transfer radical trapping, mechanisms where you can get different types of 1,2 or 1,3 difunctionalization based on the selectivity of these HAT steps that you're performing. And so we need new mechanisms to be able to tackle this type of sequential HAT mechanism. And so this is the project that I'm going to be talking about mostly today. And so one of the mechanisms that we really wanted to explore was thinking about how we can use alkynes as a linchpin coupling partner in this type of chemistry to get at a predictable difunctionalization selectivity, as well as to enable this HAT radical trap mechanism. And so The initial step that we propose in this type of chemistry is a hydrogen atom abstraction that can then generate a carbon-centered radical on an unfunctionalized or on an unactivated cyclic alkane precursor. And this, and alkynes can act as effective radical traps for carbon-centered radicals. Alkynes aren't as effective as an alkene, for example. That rate of radical addition is going to be significantly lower. But this is a known step in organic chemistry. But I think the key thing here that we were thinking about was that upon this, if you can see successful hydrogen atom abstraction and radical trapping with your alkyne coupling partner, you generate a vinyl radical intermediate. And this vinyl radical intermediate is going to be an effective hydrogen atom abstractor in its own right. It's going to be mildly nucleophilic, and it should have a predictable intramolecular HAT step that you can access. So we propose that you can access this 1,5-HAT given that you're generating a stronger CsP2H bond compared to the alkane CH bond that you're breaking. Additionally, once you do this intramolecular 1,5-HAT step, you are generating now a more reactive alkene intermediate on your compound of interest. And so we propose that once you generate this radical on your cyclohexane, for example, that this can then be trapped by the alkene that you've also just generated. Additionally, if you, through electronics or sterics, disfavor this radical addition to generate these types of bicyclic compounds, you can get acyclic radical trapping with a different type of functional group trap. And so this is a modular platform that we're proposing for accessing 1,3 difunctionalized products selectively. And some of the questions that we're interested in exploring in this type of chemistry is the effects of the functional group that's on your alkyne of interest. So as we change, for example, an electron withdrawing group here, we should be changing both the radical philicity of this vinyl radical intermediate as well as the geometry of this vinyl radical. I've drawn this vinyl radical suggestively in this bent geometry, but as you change this electron withdrawing group, the nature of this electron withdrawing group, you'll change the degree of the bent angle of this vinyl radical from being planar to being more bent. And that should affect the rate of this 1,5-HAT, intramolecular 1,5-HAT process, as well as the identity of this electron withdrawing group will affect the radical addition step as well. Additionally, we're interested in the CH partner of interest and the reactivity and selectivity effects of both the size of these cyclic rings and how this will affect these types of steps that we're proposing here, as well as we build up functional groups on these rings, how we can manipulate going from 1,3 selectivity to 1,4, 1,2 selectivity in these difunctionalized reactions. And so I have a fantastic first-year graduate student who's working on this project, and she has been able to develop a photoredox-mediated method for doing these types of difunctionalized products with unactivated cyclic alkanes and various types of alkyne coupling partners, using 4-ZZIPN as our photocatalyst of interest and quinnucleidine as our hydrogen atom abstractor to initiate this process. And so we can see that this type of chemistry works to varying degrees with a variety of unactivated cyclic alkanes and alkyne coupling partners. And so we can see we can go from a pentane ring to a cyclohexane ring to cyclooctane as well. And as we increase this ring size, we see a slight increase in yield of these reactions, And we see that we can have both difunctionalized as well as monofunctionalized alkyne coupling partners in this chemistry and see successful radical trapping as well as cyclization occurring. Additionally, when we introduce heteroatoms into these rings, we still see successful cyclization of these types of reaction partners. So as we go to tetrahydrofuran as well as dioxane, we see selectivity in these types of reaction partners. And the selectivity in these reactions is dictated initially by the quinucleidine hydrogen atom abstractor, which will abstract the alpha bond, the CH bond alpha to the oxygen atom in these heterocycle rings. And then the second degree of selectivity is dictated by the 1,5-HAT step of our alkyne coupling partner. And because we see this selectivity, this is evidence that it's going through this proposed mechanism and not simply going through a quinucleidine only mediated HAT process. And so one of the proposed mechanisms that we are evaluating currently is shown here, where we both have a hydrogen atom transfer catalytic cycle as well as a photoredox catalytic cycle that intersect. And so our photocatalyst 4-ZZIPN will be excited by visible light and to undergo this single electron process with our quinucleidine catalyst to generate the radical cation. This abstracts a hydrogen atom from cyclooctane, for example, to generate our first carbon-centered radical. This carbon-centered radical then can be trapped via our alkyne starting material to generate this vinyl radical intermediate, which performs a 1,5-HAT step to generate our second radical intermediate on our cyclooctane or cyclic alkane of interest. And this is where we propose that if we have a more bent radical in this position, we'll see more success, a more efficient 1,5-HAT process shown here. And so once we get to this radical on our cyclooctane, this can then do an intramolecular radical addition to this alkene of interest to generate our bicyclic compound shown here. And then we can get a turnover potentially of our photocatalytic cycle through another single electron transfer step. We also, we started with cyclic alkanes to be able to access these types of interesting bicyclic compounds that are widely prevalent in diterpene synthesis. However, the amount, one of the reasons why we targeted these is because generating these small ring systems, small to medium ring systems, can be quite challenging through traditional organic reactions. But we also can start with acyclic alkanes and see a successful generation of these highly functionalized 5-membered rings as well. One of the interesting observations that we've made is that if you have an incredibly activated cyclic CH partner, such as this ether shown here, we actually see generation of this aromatic heterocycle where we're generating first the acyclic compound and then we're desaturating it to generate the aromatic ring system instead of the fully saturated ring system. And so this is one of our next steps that we're going to be pursuing is using this type of chemistry to generate highly functionalized aromatic heterocycles as well. Additionally, we saw that when we started to explore other cyclic alkane coupling partners, what we would have traditionally expected as our highly functionalized 5-membered ring, we did not see at all. And instead, we saw this lactone forming. And this was very puzzling to us, since we would have expected that once we generated this tertiary-centered radical on our alkane, we would see this successful cyclization product that we have been seeing in all of the other previous reactions. And so we took out our alkyne and thought to look for if this was happening just simply on its own, and if it was a faster process than the radical addition to the alkyne. And indeed, we do see this lactone forming without this alkyne present. And so I had a fantastic, and then this is technically a formal 1,6 difunctionalization of alkanes, which is quite a challenging reaction to think about performing under traditional synthesis. And I had a fantastic undergraduate student, Eric, who took on this reaction and observed this unexpected product and decided to optimize this reaction on its own. And so he was able to look at a different type of photocatalyst and add in oxygen to increase the yield of this lactone product shown here. And so he was using tetrabutyl ammonium decatungstate as a hydrogen atom abstractor and the photocatalyst. So you can excite this, and this will act as what you can think about as an oxygen-centered radical to abstract a CH bond from your alkane of interest. And so one of the initial investigations that we have been performing in this chemistry is to look at what type of radical intermediates that we're forming in this reaction. Since we're still exploring the exact mechanism that this type of reaction is undergoing. And we've seen the tempo adduct of this tertiary radical. So we know that at least initially our tetrabutyl ammonium decatungstate catalyst is abstracting a hydrogen atom from this tertiary position. And so we've been able to develop several different methods for getting at difunctionalized products and moving away from this single functionalization strategy. And I think that this is really inspiring in terms of thinking about new synthetic routes to different types of compounds that we can use these types of difunctionalization strategies to develop more efficient syntheses of drug-like molecules. And just, I'm a new assistant professor, and so I've talked about one area of our research, which is this difunctionalization chemistry, but we have several other research areas that we're pursuing in my group. One of which is thinking about these non-covalent interactions, these lone pair to pi star interactions, where you have a lone pair donor group that can donate electron density into a carbonyl acceptor. And we're really excited about this type of non-covalent interaction to design new chiral catalysts and ligand scaffolds, and using this type of non-covalent interaction to control radical selectivity and enantioselectivity in various reactions. And we're using a lot of these catalysts to do isotope exchange chemistry and develop enantioselective hydrogen isotope exchange reactions. And another big portion of our group is thinking about nanoparticle catalysis, and specifically using quantum dots as photocatalysts. And so we're building libraries of these quantum dot photocatalysts with different ligands passivation techniques to generate, to do cross-coupling chemistry and to convert acyl chlorides to their aldehyde partners in selective chemistry. And so the first half of my talk, I would like to think was done in Abigail Doyle's group, and so I would like to thank her as well as all of the Doyle group that I was a postdoctoral fellow in. And I started at Princeton with Abby, and then we moved to UCLA throughout my postdoc. And I couldn't do any of the chemistry that I've talked about in the second portion of my talk without my fantastic group members shown here, specifically Gulenhall and Eric are the students who worked on the research projects I discussed today. And I would also like to thank our funding agencies and all of the organizers and this fantastic conference. Thank you very much, and I'd be happy to take any questions.
Thank you. Very nice presentation.
Thank you.
I wanted to ask you if you ever thought about using just acetic acid as your metal radical precursor.
Yes. So the phthalimide ester is acetic acid derived. And so you can use acetic acid to generate a methyl radical, but it goes through a different electron transfer process. So usually you'll do a proton-coupled electron transfer, which will, in your photocatalyst, will give you a reductant at the end. And so you can use that to do basically the opposite of what we're doing, which was to generate a radical and then a carbocation through an oxidative radical polar crossover to trap that and then trapping that carbocation with a nucleophile. But if you started with acetic acid, you could do a reductive radical polar crossover to close your catalytic cycle, and then you would generate a carbanion that you could trap with various electrophiles. So the answer is yes, we can use acetic acid as a methyl radical source, but you would get at different products.
I have another question.
Yes.
So you use this iridium catalyst in very low catalytic loading, like
yes.
1 mole %.
Yes.
Why it is so? What happens if you increase the loading, or it's an optimized condition?
So that's an optimized condition. You can go up to 2 or 5 mole % and see relatively similar yields. If you go past that, you actually see lower yields because you start seeing competitive photocatalytic interactions rather than the electron transfer with your substrate of interest. And so this, and also we've tried to use as little of the iridium photocatalyst as possible since it's quite expensive. We can also use organic photocatalysts. I didn't show it here, but we have organic photocatalysts as alternatives to the iridium photocatalyst that I showed that are just as effective in this chemistry, which we use in about 10 mole % loading. Usually organic photocatalysts are used in slightly higher loadings because one of the downsides of organic photocatalysts is that they can be functionalized in your reactions often. And so you see small degradations of your organic photocatalysts over time, whereas the metal photocatalysts like iridium photocatalysts or ruthenium are quite robust. And so we can take NMRs of these photocatalysts post-reaction and we see no degradation or functionalization of these photocatalysts. And so one of the things that we actually do sometimes in these reactions is re-isolate the photocatalyst, and you can purify it on a column and reuse it several times and without seeing degradation in the quality.
Thank you.
Mm-hmm.
Thank you very much for a nice presentation, very beautiful chemistry.
Thank you.
And I think the second part indeed will lead you to many new transformations in the future, I'm sure. So I would like to refer to the first part of your talk about iridium chemistry. And you said that the good match of oxidation and reduction potential is essential because you reduce your starting material to generate methyl radical, but you oxidize your intermediate radical to get a cation. But don't you think that a little bit of mismatch would be also beneficial because you can intercept the radical you produce with, let's say, a very electron-rich alkene, and which would have a lower oxidation potential that you can then oxidize even more easily.
Oh, that's a great idea. And yes, I think that's absolutely can be done, and would be a way of doing like a 3-component coupling, right? I think that would be very useful. One of the benefits, like you're saying, of this sort of mismatch as well is even if you get unselective CH abstraction, the oxidation step to the carbocation can be selectivity determining as well. So if you get a CH abstraction of a primary to get a primary carbon-centered radical, and you also get abstraction to the tertiary centered radical, you're going to get selectivity for the oxidation of that tertiary centered radical because it has a lower oxidation potential. And so you have two selectivity determining steps within your reaction, which is also a nice benefit of this dual redox gated process.
Also, in the second part, have you ever considered like trying again some benzylic substrates, like for instance orthoxylene, like to make some annulation chemistry to 6 membranes?
Yes, it's ongoing. Yes.
Also, para substituted one would be super nice because it's like cyclophane.
Yes,
A bit difficult to usually.
that might be
access.
difficult based on like the strain of the system, but that would be a fantastic example, I think. Yes.
And if I understood correctly, in that chemistry, the last step, you get electron back, right, to generate an ion stabilized by electron withdrawing group.
Correct.
I was always thinking that if you start with like a benzylic substrate, which is very electron poor, like with nitro substitute, and if you generate this vinyl radical, would it be possible to cyclize it into the ring and get Meisenheimer type, complex at the end, something like this?
Potentially,
because typically
yes.
when you do radical trapping by aromatic ring, you oxidize afterwards to get electrophilic, just to get proton release. But maybe we can do opposite polarity stuff.
Yeah, that's an interesting thought. And yeah, you see that step in other types of mechanisms, so I don't see why it wouldn't be possible in this system as well. Yeah.
Thank you.
Thank you for the questions.
So I want to ask, you use this terbutyl nitrile as a solvent.
Yes.
Do you see this radical additions to the nitrile groups?
Oh, that's a great question. No, we don't see methyl radical addition to the nitrile. We use specifically tert-butyl nitrile because it doesn't have abstractable CH bond, or it has less abstractable CH bonds than typical solvents for the type of chemistry that we're doing. And so pivalognitrile is sort of a unique solvent in CH functionalization chemistry. If you're doing any CH functionalization chemistry and you're optimizing an acetonitrile, I would try pivalognitrile because you see less competitive CH abstraction with the tert-butyl functional group compared to the methyl radical or the methyl functional group on acetonitrile. But we don't see any sort of radical addition. But if you only, under the conditions that shown here, if you add in a significant amount of water to the reaction and don't have any other nucleophile, you can get Ritter type chemistry to occur.
Thank you.
Mm-hmm.
I also have one question about alkynes, maybe other substituents. Have you thought of using?
Yes. So this is, we're building a sort of a library of alkynes for this difunctionalization chemistry with varying functional groups from electron donating to electron withdrawing groups, and the di-substituted variants of this as well, because we've seen that this is leads to the biggest differences in the types of products that we get out in these reactions, as well as the rate of the reaction steps. And so one of the key functional groups differences that we're evaluating is going from a phenyl group substituted alkyne, which will give a very linear vinyl radical intermediate, to a nitrile substituted alkyne, which should give a more bent vinyl radical intermediate, as well as an ester, a diester, which I think I showed one example of shown here. And one of the things that we're interested in that specific type of functionality on the alkyne is that when you generate that vinyl radical intermediate, you actually have a lone pair to pi star interaction that you see from one ester substituent to the other that traps it in a cis conformation of that vinyl radical. And so you have a more directed 1,5-HAT step because you have a more bent radical geometry in that step. And you also then slow down that radical addition process because you're trapped in that cis alkene conformation. And the cis alkene will be less planar than the trans alternative. And that has a slower radical addition rate as well. So there's many different effects that we're exploring in this chemistry based on the functionalization on our alkyne intermediate.
Are you thinking about intramolecular cyclization then, not only between two?
Correct, yes, yeah. So we're hoping that if we can control the radical addition based on the substituents on our alkyne, that we can go from intramolecular to intermolecular.
Okay, thank you.
Maybe you can accept the third time this alpha radical and do the alpha hydrogenation.
Yes, hopefully, yeah.
I have one more question.
Yeah.
When you have this 20-30% yield, so what happens with the starting material?
Yes, so the lower yields that I showed here were based on the time point that we were getting at. So the longer we run these reactions, the higher the yields we get, but we're trying to optimize that reaction time down currently. One of the some of the mass balance that we see in the reaction is the alkene product. So we're getting unselective HAT in this step, and so depending on the CH partner, that will dictate how competitive we're getting for one intramolecular 1,5-HAT versus HAT from the solvent or another equivalent of alkane in solution. And so we see dependence on the equivalency of between our CH partner and our alkyne partner will dictate how much of the alkene intermediate we get at the end. But one of the benefits of this is that if you just let the reaction run longer, you can convert over time that alkene that you get to desired product via another quinupridine HAT step as well. But that's a much slower process, and so we're trying to optimize away from simply just letting the reaction run for a few days so looking at ways to prevent that unselective HAT from the vinyl radical intermediate.
Thank you.
Thank you very much for your presentation, and you may proceed.